Okay, this section we're going to cover is on the anterior pituitary. It is week 4, and there's a lot that we need to cover, so we're going to get started. When we talk about the anterior pituitary, the structure itself is more glandular than neural, and you can see that under a microscope. The anterior pituitary regulates production and release of tropic hormones. The anterior lobe is also referred to as the Adeno Hypophysis. The primary hormones secreted include luteinizing hormone, follicle-stimulating hormone, growth hormone, adrenocorticotrophin hormone, prolactin, and thyroid-stimulating hormone. Now, there's a variety of others that play a minor role, and I will include those. Endorphins, lipotropin, and melanocyte-stimulating hormone. When we look at the hormones released from the anterior pituitary, they have somewhat similarities. And so they're put into 3 classes. Glycoproteins, which have a similar structure. They usually have like an alpha subunit chain and a beta subunit chain. And the beta subunit makes it unique. So you see TSH, LH, FSH, and interestingly enough, also HCG, which if you're not familiar with, that's human chorionic gonadotropin. That's the hormone we use as a diagnostic of whether a woman is pregnant or not. Okay. The next grouping is pro-opiomelanocortican, POMC. Now, from POMC, which is a much, much larger peptide chain, you get ACTH, beta endorphin, alpha, beta, and gamma melanocyte stimulating hormone. These are all controlled by post-translational cleavage. In other words, the POMC is first made, and after translation occurs, then you have it chopped up into smaller pieces. Now, the third group, growth hormone and prolactin, interestingly enough, are structurally similar to human placental lactogen. When we look at anterior pituitary function, we have a variety of feedback controls. Most of them are almost all of them actually, are really negative feedback. And environmental factors may increase or decrease pituitary activity by increasing or decreasing hormone secretions from the hypothalamus, which then acts on the pituitary gland, which then releases its stimulating hormones or trophic hormones to the various target glands and tissues. The pituitary hormones may also inhibit their own secretion by a short feedback loop back to the hypothalamus, as well as the target gland hormones may also have a negative feedback to both the pituitary gland and the hypothalamus. Now, when we look at cell types, what we're going to do is compare the hypothalamic factors, the pituitary hormone produced. We have basically 5 major pituitary cell types: lactotrophs, corticotrophs, thyrotrophs, gonadotrophs, and somatotrophs. The hypothalamic factor that stimulates lactotroph is dopamine, and this causes the production of prolactin. The hypothalamic factor CRH acts on corticotropins, and that creates POMC, which is then broken down to ACTH, beta lipotropin, alpha MSH. Also may include also beta MSH and gamma MSH, but also what's known as beta endorphin. That's an endogenous opiate, which we'll mention a little bit more later on. TRH acts on thyrotrophs, and that causes the pituitary to release thyroid-stimulating hormone. Gonadotropes are triggered by the hypothalamic factor GnRH, and that causes the gonadotropes to release LH and FSH. Now, GHRH triggers somatotropes to produce growth hormone. And as you can see here, there's a big difference here between the posterior pituitary and the hypothalamus. The hypothalamus here will have relatively shorter axons, which will then release their releasing hormones into this initial capillary network, which will travel down the portal blood vessels to the second capillary network here in the anterior pituitary. It is in here that the anterior pituitary cells are going to release their various hormones, these trophic hormones. And as you can see in an overview, you can get quite detailed in how the pituitary hormones act on so many other aspects of the body, not merely just particular organs, such as the adrenal gland and thyroid gland, but also various tissues in the mammary glands. You will have also action by various other cells like melanocytes, as well as the testes and the ovaries. All of the anterior and posterior pituitary hormones bind to their membrane receptors and use, interestingly enough, secondary messengers. That's the release of cyclic AMP. This is why, in part, when you first were hearing about the pituitary, a lot of scientists would call it the master gland. Later, that was revised when we found out that there were hypothalamic releasing hormones. And so the hypothalamus really became renamed as the master gland. Okay, now a couple of things you need to be aware of also. That's the cell signaling pathways. The hypothalamic peptides to the pituitary cell receptors, if we were to look at TRH and GnRH, they're all G protein coupled receptors that act to increase the activity of phospholipase C. This, of course, is going to result in an increase in IP3 and DAG with an increase in calcium ions intracellularly. CRH and GHRH are also G protein coupled receptors, but they tend to activate or increase adenylate cyclase activity, which is going to result in the increase in cyclic AMP. Now, somatostatin and dopamine, which play a role in triggering the anterior pituitary, they also play a role with a G protein coupled receptor, but this is different. They're going to basically decrease adenylate cyclase activity, thereby they're going to decrease the amount of cyclic AMP production. You want to also be aware of this. Now, PRH. We know that dopamine has inhibitory effects to the release of prolactin. And every once in a while, you'll hear individuals talking about sort of prolactin releasing hormone. There's no real certainty of that. As a matter of fact, some scientists would assert prolactin is its own stimulus for release of even more prolactin. Part of this is because dopamine is really also called prolactin inhibiting hormone, but no one has been able to successfully isolate the PRH. Now, the pituitary peptides to the target organ receptors, what do we got here? Well, TSH, LH, FSH, and ACTH, again, are all G protein coupled receptors. They will activate adenylate cyclase, which will result in an increase in cyclic AMP. Now, earlier diagrams showed you some saying all, and unfortunately this doesn't always happen. In GH and prolactin, you have a class 1 cytokine receptor, pardon the spelling, and this will activate the protein kinase C. This results in an increase in kinase activity and phosphorylation of proteins. And what you can see here in this diagram, and it may get somewhat overwhelming, is really just all of the hypothalamic hormones that you see. GHRH, TRH, CRF, they call it the factor, but it can also be the hormone, CRH. As I said, all we have here for prolactin is at least solidly is dopamine, and that's an inhibiting factor, and then GnRH. These will all act on the anterior pituitary. Now, if you notice right here, GnRH is going to cause the release of two hormones, FSH and LH, follicle-stimulating hormone, luteinizing hormone. These will be targeting the gonads, ovarian follicles, testes. We'll get into what they do in a bit. Prolactin will, of course, act on the mammary gland and milk production. ACTH acts on the adrenal cortex and the subsequent production of glucocorticoid release. We'll get into a little bit more in detail about that. TSH acts on the thyroid, and you get T3, T4. Endorphins, interestingly enough, act on pain pathways, with the result being an inhibition of the perception of pain. Growth hormones were going to act on a variety of tissues, including bone and muscle, and promoting growth. Now, a couple things to keep in mind. Notice that there's even a minor spelling error here too. But the more important point is this. The anterior pituitary is referred to as the Adeno Hypophysis. The posterior is the neural hypophysis, because the posterior, you can see, is more neural in structure. The anterior is more glandular in structure. Every one of these hormones has a site of action, either target tissue or a broad target tissues, a particular organ such as the thyroid or the adrenal cortex or the gonads. The interesting thing is we really haven't gotten anything clear about beta-lipotropin, and I'll explain more of that in a short time. So what we're going to do now is we're going to go into a couple of pointers about each of the different hormones released by the anterior pituitary. First, thyroid stimulating hormone, TSH. Its target organ is the thyroid, and it's involved in controlling the release of T3 and T4. TSH will bind to a G-alpha S protein. This is the protein-coupled receptor in the thyroid, activating adenylate cyclase. You're going to see an increase in cyclic AMP. It's interesting to note also that protein kinase A, that signaling pathway, is also activated. This is controlled by negative inhibition of the thyroid hormones, especially T3. So T3 will have a negative feedback on the release of TSH. As you can see here, we have the anterior pituitary, TSH acting on thyroid. Free T4, interestingly enough, seems to play a role on the pituitary release, whereas you have T3 converting over to T4 for the hypothalamic inhibition. Now, by the way, this is the shape of actually TSH, and it's a relatively small peptide. But the other thing that's very important about this, aside of the fact here's our receptor, here is the process to activate cyclic AMP, is that you can also fake for thyroid cells the signal of TSH, and that is by TS antibodies. They will bind to this receptor. Also, there has been some documented influence by HCG. Again, this is the hormone that's involved that begins to spike upwards after fertilization has occurred. Now, here's something also to keep in mind. How do we define hypothyroidism versus hyperthyroidism? Usually, hypo's pretty clear. You have an underactive thyroid gland. Hyperthyroidism, you have an overactive thyroid gland, but also that's why doctors not only do tests to look at blood levels of T3, T4, but they also use the analysis of TSH. Because if you have a high TSH but no T3, T4, that's one way of determining whether it's purely a thyroid. Or if you have a high low TSH levels and an overactive thyroid, there may be other factors that are fooling the receptor to overproduce T3, T4. We'll get more into the thyroid in a couple of weeks. The next one we want to deal with are the gonadotropins, luteinizing hormone and follicle stimulating hormone. Most gonadotropes produce both LH and FSH. There are a fraction of the cells that will only produce one. And sometimes what happens is, depending on the stimulation, they will be producing one where they normally produce two. So they flipped over. Both of these exert physiological effects on the gonads, in other words, testes and ovaries. So on the testes, LH stimulates testosterone production from the interstitial cells. These are referred to as Leydig cells. FSH stimulates testicular growth and production of androgen-binding protein from the Sertoli cells. This will eventually enhance sperm maturation. That's for the testes now. What about the ovaries? FSH stimulates ovarian follicles to grow, triggering releases from follicular cells. Now, as you have a rise in estrogen, this feeds back to the pituitary and triggers a reduction in FSH and a rise in LH release. Why? Well, LH triggers the release of the egg from the ovary. In other words, this is the ovulation step. It also will stimulate progesterone production from the corpus luteum, the remnant of cells from that ovulation that are still present on the ovary. Here is one example. You can see here the pituitary produces LH, acts on the Leydig cells, FSH works on the Sertoli cells. This leads to maturation of sperm. This leads to the synthesis and release of testosterone. Testosterone has a negative feedback on both pituitary and hypothalamus to reduce GnRH as well as FSH and LH. Now, in a menstrual cycle, take a look at this. You see where estrogen rises and then drops. And you see also where, as it's beginning to drop, you get this massive spike of LH, and then it drops off. Following that spike, you get this increase in progesterone. Ah, yes, but what about FSH? FSH, about the time that you're having ovulation, spikes and then falls off. Okay? We'll get more into the reproductive side in some weeks ahead. Now, feedback control. Both of these hormones are controlled by absences of GnRH as well as negative feedback from the target organ hormones. So in other words, you're going to have testosterone, estrogen, and then another hormone, which is referred to as inhibin, which will feed back. Inhibin is particularly seen with the females, and what you see here, inhibin A and B, they're going to reduce the FSH secretion. There is a positive feedback from estradiol at the mid-cycle. This is going to enhance or boost LH, and yet it will inhibit FSH. In females, estrogen has a negative feedback to the hypothalamus and the pituitary. In males, they do produce inhibin, and it has a negative feedback control on the hypothalamus and the pituitary. When you're talking about this, then you're talking about the entire bailiwick. GnRH, FSH, and LH. And as I've noted before, here is the male side. We usually go much more in depth with the female, but I wanted to just cover both of them, LH and FSH here. You can see where these are working on the testes cells, and interestingly enough, the Leydig cells will have a positive feedback effect on Sertoli cells to facilitate the spermatogenesis. Inhibin is released by Sertoli, which provides a feedback to the anterior pituitary, whereas testosterone can do a negative feedback on the anterior pituitary as well as the hypothalamus. Now, here you see this. Now, what are the granulosa and thecal cells? These are the cells that are involved in the formation of the egg cell. So, FSH will work on granulosa cells. That ends up producing inhibin, which will feed back to the anterior pituitary, estrogen. Now, with the thecal cells, LH runs those, which leads to the production of androgens, which are then converted to estrogen. Estrogen has the dual pathways of negative feedback working on both anterior pituitary and hypothalamus. And this is a little neater, as you can see. And what we end up with is thecal cells produce the androgens, which are converted to estrogen. They will also be converted into, and the thecal cells will start taking over progesterone production. When you look at progesterone, this is going to be sort of after ovulation. Beforehand, estrogen is, shall we say, running the show a lot. Okay? Progesterone, this is for endometrial development and the mucus plug that occurs at the cervix. If you look over here, you have FSH and LH, and they are directed at several different levels. Both of them play a role in gamete production for the ovaries. And it's the steroid and peptide hormones, inhibin, as well as estrogen, and to a lesser extent, progesterone, that have the negative inhibitory effect. Now we get to POMC, pro-opiomelanocorticin. This is a production cell. This is a production that occurs in the cell of the corticotropes. They possess receptors. They have CRH1 and CRH2. CRH1 is really the POMC synthesis trigger. You're going to see a rise in cyclic AMP, the secondary messenger. The function of CRH2 is not really certain. POMC is post-translationally cleaved into ACTH, beta endorphin, which is an endogenous opiate peptide, alpha, beta, and gamma melanocyte stimulating hormone. Okay? Now, if you look at this, here's the big POMC gene. It gets reduced, and this is before even translation. So you have this reduction of some of the introns, and you now have your messenger RNA. The messenger RNA gets made into POMC. But then you start having basically POMC broken down into several different units. ACTH is one, beta-lipotropin is another. In the brain, these products may get further broken down. Oh, by the way, down here, this metenkephalin is similar to beta endorphin. It is another type of endogenous opiate. But if you notice here, alpha MSH, gamma MSH, which is kind of iffy, and beta MSH, we're going to get into. All of these are involving the melanocytes. ACTH, there's sometimes what they call the clip is removed. And as you can see here, you also have lipotrophin, going from beta to gamma. Now we can see this in another way too. I just wanted to give this as several different demonstrations. Because what we find is that, are we talking about purely just the anterior pituitary, or does some of this actually get synthesized in other brain tissues? The answer, it does get synthesized in other brain tissues and gets reduced post-translationally. And this is where we get these unusual peptides that may or may not have a lot of influence. We'll discuss them just a little bit more in a second. So here we have again transcription, polyadenylation, transduction. You go through the processing of this. In the beginning, you clip off the beta lipo hormone. And what happens is this gets broken down to gamma lipohormone, and you also have lipotropin, excuse me, and you end up with a beta endorphin here. If you notice, you keep having these clips here, and some of them end up with viable, useful hormones, such as MSH, and both gamma and alpha. ACTH is the big concern because that is going to run what is essential for how we turn on basically the adrenal glands. So let's talk about this. ACTH is adrenal corticotropin hormone. The release is stimulated by psychological and physical stress, fear, infection, hypoglycemia, surgery, trauma. The half-life in the blood is 3 to 4 minutes. It is considered critical for mediating stress and the adaptive response to stress. It is released in very small quantities. The circulating levels average anywhere from 2 to 19 picomoles per liter. It is interesting to note that this is released in pulses, with the highest amount at 4 a.m. in the morning. Lowest is during the daytime. Now, ACTH binds to a G-alpha S protein, which is coupled with the MC2R. This is a receptor, and this is located in the adrenal gland cortex. It activates adenylate cyclase, which will increase cyclic AMP, as well as activate protein kinase A. The effect is going to trigger the production and the release in the adrenal gland of glucocorticoid, which we also call cortisol, and yet in a lesser degree, we're going to get a release of some of the mineralocorticoids, aldosterone. Now, we'll get back to the adrenal gland later on in the course. Aldosterone is definitely different in the sense that, although they're both steroidal, one is going to be very useful for dealing with stress and managing glucose levels, and the other is going to be more involved with the maintenance of mineral, particularly the electrolytes in our blood. Now, it's interesting to note also that the release of cortisol has the same rhythmicity as ACTH. So basically, when ACTH is at its peak, what you're going to see is not too soon afterwards, maybe minutes, that you're going to have this peak of cortisol. You also have feedback inhibition of both ACTH and CRH via cortisol. And this will bind to the glucocorticoid receptors in the hypothalamus and anterior pituitary. Now, it's also interesting to note, ACTH will bind to a lesser degree to the MC1R receptor. This is the melanocyte stimulating hormone receptor that is located on melanocytes. This can still cause the production of melanin granules, and it can trigger a bronzing of the skin in Addison's disease. Now, I don't want to jump too far ahead in Addison's disease, but to give you just a pointer, Addison's disease is when you have no production of cortisol, but you've got a tremendous quantity of ACTH being present. As you can see, ACTH will function, stimulate in the cortex cells, an increase in cortisol to pregnenolone, which from there will be converted into adrenal steroids. It will also promote the growth of adrenal cortical tissue. And as you can see here, we have a couple of demonstrations here of the negative feedback. Particularly, it's mostly just cortisol. The shorter loop is going to work on the pituitary to reduce ACTH, but the longer loop is going to have a negative feedback on CRH secretion. All right? Cortisol you want to keep in the back of your mind when we get into that. This is very important in managing stress and helping to deal with things like inflammation, etc. More later on that. Now, the melanocyte stimulating hormone, MSH, has receptors MC1R, MC2R, and MC5R that all have roles in the skin. Now, there are other receptors located in other parts of the body. The MC3R, though, its function is really unclear. MC4R, that happens to be located in the brain. And it's interesting, it has effects on behavior, feeding, and appetite regulation. Okay? So what was getting to be sort of blurry was, well, what do these hormones do if there's not binding in this part of the body or that? It may feed back into and play a role in certain aspects of behavior. So what do we have? MSH will stimulate the rapid translocation of melanosomes, these are the melanin granules, in dermal melanophores to affect a rapid color change. Now, melanogenesis in normal and abnormal melanoma epidermal melanocytes also. So what I did was I threw together these nice little amino acid sequences. Really, these alpha, beta, and gamma are really human. They just had this comparison with porcine. But what you can see is there are different links for each of the different types. And that corresponds to the post-translational modification that occurs with POMC. And as you want, you can take a look here. Here's the information about melanin synthesis. You have the melanocyte stimulating hormone, which will bind to melanocortin 1 receptor. That's the MC1R. It'll act on adenylate cyclase, causing an increase in cyclic AMP, which activates tyrosinase. And you see, the idea is that tyrosinase is really going to put together two tyrosines to make melanin. And what you have, you can see right here, they're going to transfer these granules, these melanosomes, they will be eventually impregnating themselves into keratinocytes. You can see the chemical reactions here and what happens. You have tyrosinase that will make basically the dopa, which will then make the dopaquinone, and then you end up with making either pheomelanin or uromelanin, which eventually contribute to melanin itself. Again, here are the different sequences that you see of ACTH and MSH. And some of these others, gamma 1, gamma 3, these are partial analysis. It's entirely possible you're going to see more of these neuropeptides, such as MSH, being understood from both the behavioral side as well as the physiological side. Now let's get to beta-lipotropin. That's LPH. This is a little bit of conundrum. ACTH and beta-lipotropin are synthesized as one molecule, then they're split before release. What we do know of their functions is that they promote the release of fat reserves from the liver into the bloodstream. It is reported that LPH will perform lipid mobilization functions such as lipolysis and steroidogenesis, this usually occurring most in the liver. Unfortunately, no receptor has been ever located for LPH. Now we get to beta endorphin. This is an endogenous, meaning self-made by your body, opioid neuropeptide. And it's a peptide hormone that's produced in certain neurons within the central nervous system, as well as the peripheral nervous system. What is its function? It's been known to be associated with hunger, thrill, pain, maternal care, sexual behavior, and reward cognition. So if you're sitting there going, thrill, what do you mean? This is behavioral effects. It's primarily used by the body to reduce stress and maintain homeostasis. Interestingly enough, it has a high affinity for and lasting effect on the mu opioid receptors. The function includes also negative feedback inhibition of GnRH release. As you can see here, if you had a presynaptic neuron and you have an endorphin receptors here, they're going to send over and shut down the release of another nociceptive neurotransmitter called substance P. And what happens is you don't have a sense of pain in the brain. Okay? Now, is it easy to make? Is it easy to get as an analgesic? No. There have been attempts at trying to synthesize it using, let's say, right-handed as opposed to left-handed amino acids included, but nothing has come to market to which I am aware of to this date. Now let's get into one of the biggies, growth hormone, GH. It is 191 amino acid peptide. It's made by the somatotroph cells. These are growth hormone is structurally similar to prolactin and to chorionic somatomamotropin. That last one is placentally derived, meaning that you find it only in the placenta. Now, GH is released through pulsatile bursts. The regulatory control is that GHRH stimulates GH release. And interestingly enough, somatotropin inhibits GH release. We're going to talk a little bit about somatotropin. Somatotropin is sort of one of the controllers. It has a negative effect. Also, one of the other interesting things is that insulin-like growth factor, IGF-1, inhibits GH release via enhancing somatotropin release. We're going to get more into IGF-1 in a few minutes. Another stimulus will be low blood glucose concentrations. GH may directly inhibit the somatotroph release of GH. This is an autocrine influence. GH acts two ways on body growth, directly and indirectly. So let's start off with directly. It will act on bone to increase bone and cartilage formation. It will act on adipose tissue and stimulate release and oxidation of free fatty acids. Skeletal muscle stimulated the amino acid uptake and incorporation into protein. We also will have cell proliferation, and it also suppresses protein degradation. Remember, bones are made up of not only just the mineral hydroxyapatite, which is calcium phosphate, but also the scaffolding, which happen to be collagen fibers that help support it. Now, the liver gets stimulated by IGF production and releases, the release of GH will promote gluconeogenesis and reduces glucose uptake. What I'm saying here is that GH will act on the liver, which will cause the release and the increased production of IGF-1. Also, it will stimulate hepatocyte glucose output. GH will also enhance in the immune system the functions of T cells, B cells, natural killer cells, NK cells, and macrophage activity. In the central nervous system, the effects of GH will be by modulating mood and behavior. And finally, metabolism. GH will promote lipolysis via inhibition of lipoprotein lipase. It will decrease skeletal muscle glucose utilization, increasing lipolysis and increasing hepatic glucose production. It counteracts the action of insulin on lipid and glucose metabolism. Now, that said, you can see this here, where you see this. Now, Gilrean, we'll talk about probably in a couple of so many weeks. This has been best known as something found in the stomach, but let's take a look and focus first on GH. It will act on the liver. It will cause the release of IGF-1. It will act on things like kidney, cartilage, bone. It will also enhance the function of a variety of the other tissues. It's interesting to note that you're going to have negative feedback both at the hypothalamus and by the pituitary to blunt GH release. But let's talk about IGF-1, insulin-like growth factor type 1. It's also known as somatomedins. It is a relatively small peptide hormone produced primarily in the liver in response to GH stimulation. It's transported to other tissues to act as an endocrine hormone. Its half-life, though, is somewhat longer, 15 to 20 minutes, and it's transported either free or bound to several specific proteins that will enhance the half-life. Insulin-like growth factor binding proteins, IGFBP, yes, that's a mouthful. There's 6 different types, and they regulate availability of IGF to the target tissue receptors. IGF-1 binds to IGF receptors in various tissues with mostly anabolic effects, in other words, building effects. It will mediate anabolic and linear growth. Therefore, it promotes the effect of pituitary growth hormone. It promotes bone formation, protein synthesis, glucose uptake in muscle cells, neuronal survival, and myelin synthesis. The last 2 are important because really what it's doing is it's helping with the growth development of neurons and myelination. IGF-1 acts as a mitogen, that is a substance that induces mitosis. Therefore, it's going to stimulate the mitosis, and to do that, it's going to enhance or stimulate DNA, RNA, and protein synthesis. Now, this is all extremely essential for embryonic development. And as you can see here, we have the release from the liver of IGF, muscle, cardiac development. It's also going to act as a vasodilator. We're going to work on all sorts of factors on the brain, being both anti-inflammatory, enhancing neural development, myelination, and protection of the brain. We're going to see also normal bone growth, as well as reproductive organs that are going to basically go through maturation, the testes as well as the ovaries. It is also going to play a role in placental growth and development, as well as fetal growth and differentiation. We also have to keep in mind that IGF-1 plays a key role in glomerular development, both the function and integrity of the glomerulus. Finally, this has sort of an immune component to it in the sense that IGF-1 plays a role in thymus development and function, hematopoiesis, and immune cell function. So you want to keep that in mind. Now let's go back to a troublesome little hormone, somatostatin. It's also known as growth hormone inhibiting hormone, GHIH. I say it with a little bit of tongue-in-cheek there about troublesome. Really what somatostatin does is it inhibits the release of growth hormone, prolactin, and TSH in the pituitary. It will also inhibit insulin and glucagon release in the pancreas. We're going to get into the pancreas later on, but one of the things you're going to definitely see is that the pancreatic islets are made up of different cells that release different hormones. Insulin, glucagon, somatostatin, as well as pancreatic polypeptide. Okay? But enough of that right now. What you have to keep in mind is that peptide hormone produces in various brain regions, especially the periventricular nucleus, the arcuate nucleus, and the ventral medial nucleus, which are all in the hypothalamus. This hormone will bind to G-alpha inhibitory G-coupled proteins, resulting in decreased activity of adenylate cyclase and cyclic AMP. Okay, continuing. What you have is that somatostatin binds to a little bit different of a G-coupled protein. It's going to bind to a G-alpha I. This is going to result in the decreased activity of adenylate cyclase. So you're going to expect that cyclic AMP is going to decrease, as well as intracellular calcium concentration. So basically, this is sort of the hormonal brake on many different hormones. Somatostatin receptors are modulated by various hormones and by the patient's nutritional state, which is an interesting fact too. Here is somatostatin, a relatively small peptide. Notice that you have between the cysteines a disulfide bridge. Notice all the organs that can be affected by somatostatin: adrenals, testes, gut, pancreas, brain, pituitary, and thyroid. Finally, we're coming up to prolactin. Now, prolactin is a polypeptide that's synthesized by lactotrophs. Lactotrophs will increase in number with elevated estrogen levels, especially during pregnancy. Prolactin levels usually are higher in females than males, and the role of prolactin in males is not really well understood. Plasma concentrations are highest during the waking hours and lowest during the waking hours. I stand corrected there. Plasma concentrations, interestingly enough, are highest during sleep and lowest during the waking hours. It's interesting to note that prolactin's release is triggered by dopamine from the hypothalamus. The release is inhibited by somatostatin and by another neurotransmitter called GABA, which is gamma-aminobutyric acid. Dopaminergic inhibition of lactotroph release of prolactin is mediated by the dopamine D2 receptor, which is a G-alpha 1 inhibitory, that is, G-coupled protein receptor. Now, there are several prolactin-releasing factors found: TRH, oxytocin, vasointestinal peptide, and neurotensin. Prolactin regulates its own secretory secretion, really. Prolactin binds to prolactin receptors in dopaminergic neurons in the hypothalamus, and so this results in a release of dopamine. It's almost like it seems like a positive feedback in a way. But prolactin receptors are found in the mammary gland and ovary, as well as several regions of the brain. The effects are the stimulation of growth and development of the mammary gland, synthesis of milk, and the maintenance of milk secretion. Prolactin stimulates glucose and amino acid uptake and synthesis of milk proteins, particularly beta-casein and alpha-lactalbumin, the milk sugar lactose, and milk fats by the mammary epithelial cells. It also, interestingly enough, modulates reproductive and parental behavior. As you can see here, we have a situation where we have the lactotrophs here releasing prolactin. It will feed back in a negative sense back to dopaminergic neurons. The suckling stimulus is also acting on dopamine neurons. Sleep, sex hormones, orgasm, and stress are positive stimulus for the dopamine to be released. And interestingly enough, stress and cannabinoids have a negative effect on the stimulus to release prolactin. Now, it should be also noted that the structure of prolactin is similar to that of growth hormone and placental lactogen. The molecule is folded due to the activity of 3 disulfide bonds, and you can kind of see them a little bit. It's a little harder to see in this diagram. The non-glycosylated form of prolactin is the dominant form that's secreted by the pituitary gland. So now what we're going to do is explore the diseases of the anterior pituitary. We're going to cover a couple of different situations. Hyperprolactinemia, hypopituitarism, growth hormone excesses, growth hormone insensitivity, which is referred to as Laron syndrome, and pituitary adenomas. When we talk about hyperprolactinemia, this is excessive prolactin, which can cause the production of breast milk in men and in women who are not pregnant or breastfeeding. Now, in women, too much prolactin can also cause menstrual problems and infertility, that is, the inability to get pregnant. In men, it can lead to lower sex drive and erectile dysfunction. The symptoms for women would be infertility, irregular periods, change in menstrual flow, pause in menstrual cycle, loss of libido, lactation, that's referred to as galactorrhea, pain in the breasts, and vaginal dryness. Now, with men, well, abnormal breast growth, that's gynecomastia, lactation, yes, the men are actually giving milk from their breasts, infertility, erectile dysfunction, loss of sexual desire, headaches, and visual change. Now, if it gives you a cluing in, you're going to see a couple of times with some of these pituitary dysfunctions, headaches and vision change. Keep in mind that the optic chiasm is very close to the pituitary, and any growths that seem to spring up from the pituitary may start having an effect on the optic chiasm and thereby on vision. Here's the individual who has a headache. Here's the individual who suffers hyperprolactinemia. And you can see in here that the regular system that's producing the prolactin, they've actually done an MRI here, and you can see this larger mass here. Okay? So the causes of prolactinemia, stress, pituitary tumors are most common. Hypothyroidism can occur. But some of the signs of hyperprolactinemia, breast milk secretion, decreased libido, irregular menstrual periods. Now, the next one we're going to look at is hypopituitarism. This is a rare disorder in which your pituitary fails to produce one or more hormones or doesn't produce enough hormones, period. Depending on the loss of a particular hormone, the symptoms may appear like diabetes insipidus or ADH deficiency. But if you look at some of these specific deficiencies, here's what you're going to see. For ACTH deficiency, you're going to have severe fatigue, low blood pressure, which may lead to fainting, frequent and prolonged infections, because they're not producing cortisol to handle it, nausea, vomiting, and abdominal pain, as well as confusion. With TSH deficiencies, that can lead to fatigue, weight gain, dry skin, constipation, sensitivity to cold, or difficulty staying warm. We'll get more into the thyroid shortly. With GH deficiency, you're going to have fatigue, muscle weakness, changes in body fat composition, lack of ambition, social isolation. In children, you're going to have a reduction in growth. With LH and FSH deficiency, in women, the deficiency decreases egg and estrogen production from the ovaries. In men, the deficiency will decrease sperm and testosterone production from the testicles. Women and men may experience lower sex drive, infertility, or fatigue. In children and adolescents, there will be delayed puberty that is usually the only symptom. And here we have hyperpituitarism. Keep in mind that it could be a little bit more profound from the sense that it would not be necessarily a primary pituitary tumor, but it could be something such as the interruption of the pituitary stalk, the damage to the infundibulum. Okay? And what we have in certain cases with hypothalamic disease can include such things as exogenous steroid use, extreme weight loss, exercise, or systemic illness. Okay, this is where we're having a distortion in the production of certain steroids, such as from ACTH, etc. Now, in hypopituitarism, what you're going to probably see, all the problems of primary Addison's disease, except the dark skin, all the problems of primary hypothyroidism. They may also be present as a failure of lactation, diabetes insipidus, and growth failure in kids. You will have loss of libido, amenorrhea, weakness, muscle, and soft tissue wasting. It is possible to also have weight loss and personality changes. If you think about it, keep in mind that the pituitary is surrounded by the sella turica. It's the bony sort of saddle that the small organ is present. You can have an infarct where you have loss of blood supply to that, or an empty sella, which means that basically the tissue is never there, or an adenoma that is putting pressure on the pituitary gland and thereby interrupting either blood supply or secretion or both. Now, if we look at this, with the decreased function, we have hypopituitarism. Okay, so we can have all these different ones. The big ones I want are the pituitary dwarfism, but you can also have hyperpituitarism. And usually we're talking about disorders that either increase prolactin release or growth hormone. And growth hormone, that would be acromegaly and gigantism. So let's talk about that. Now, acromegaly and gigantism, what's the difference? It's when they are initiated. Okay? Acromegaly is an abnormal growth of the hands, feet, and face caused by overproduction of growth hormone by the pituitary gland. Now, the big deal here is, why don't we call it that in children? Because the children's epiphyseal plates have not closed on the long bones. Okay? Once you're an adult and the epiphyseal plates have closed, and you start having this larger amount of growth hormone occurring, then you're going to see what they call tower skull, lantern jaw, etc. In other words, outcropping of the jaw, an increase in the cranial structure, etc. Usually this is caused by a non-cancerous tumor. Middle-aged adults are most commonly affected. Okay? Gigantism is the abnormal growth due to an excessive growth hormone during the childhood. So the child may grow much, much taller before the epiphyseal plates close. If they still have production at that point, then they're going to be faced with acromegaly over time. The symptoms are, of course, abnormally tall stature, abnormal growth of face, hands, feet, thickened facial features, irregular menstrual cycle, excessive perspiration with slight activity, delayed puberty, double vision, and deafness. Now, if you're thinking about this, what do you mean by that? Double vision, again, is a disruption around the optic chiasm. The deafness can be because of the extreme growth in the skull that will literally crush out the inner ear structures or the nerves leaving the inner ear for noise processing. Here is an example of acromegaly. Visual field defects, prominent supraorbital ridge, in other words, kind of above the eye, large nose and jaw. Teeth are beginning to be separated, okay, because of so much growth. The jaw is growing, but the teeth are not going to be next to each other. They're going to be actually spacing apart as time goes on. Abnormal glucose tolerance test. They will have glucosuria or polyuria. Spade-shaped hands and feet, arthrosis, peripheral neuropathy, sexual dysfunction, cardiomegaly, hypertension, galactorrhea, which is usually due to an increase in prolactin, and hypertrophy of sweat and sebaceous glands. To locate them, usually you're going to get a CAT scan or an MRI. By the way, this, cardiomegaly. When you have someone who is, 6.5, 7, really going into 7.5, 8 feet, you don't hear them lasting to their 80s and 90s. A lot of them who are very, very tall individuals die earlier in life, usually because of cardiovascular disorders, because the heart has to strain to take care of a much larger body mass. Here you can see the spade shape of the hands. You can also see somewhat the curvature of the back. You may also see some problems with glucose tolerance. Here is an individual, you see the enlarged jaw, okay? And this is all due to increased GH production because of that pituitary adenoma. This would be, for this adult, acromegaly. Here's another example of this, as you can go right down through it, barrel chest, cardiomegaly, and of course, when you're having this enlarged heart, you're going to have also hypertension. You're also going to start seeing not only increased size of hands and feet, but you'll begin to start seeing degenerative arthritis because the body frame cannot withstand the much larger body mass. Okay? Finally, we get into Laron syndrome, which is a glucose hormone insensitivity. This is due to deletions or mutations in the growth hormone receptor gene. This is going to result in a failure to generate IGF-1 and IGFBP3, okay, in response to growth hormone stimulus. This is an autosomal recessive disorder, meaning that you have to have both copies present. What you're going to see for symptoms: short stature. Now, when I say short, I'm talking 4 to 10 standard deviations below the median height. Obesity, cranial facial abnormalities, micropenis, low blood sugar, and low serum IGF-1 despite elevated basal serum of the growth hormone itself. Here we have a whole family passing on several generations of this congenital abnormality of the growth hormone receptor. That's Laron dwarfism. In normal growth, you're going to have this situation where growth hormone is going to attach to receptors, which in the liver will release IGF-1. This will eventually be a negative feedback for the growth hormone. Problem here is you will have growth hormone released and actually be present in much higher amounts. Excuse me. But the mutated growth hormone receptors will not pick up the signal, and therefore you're not really going to get a lot of IGF-1 at all. It's going to lead to dwarfism, obesity. Now, some people talk about cancer protection or an anti-aging signaling. That's debatable. What we're talking about is that certain cells won't feed on growth hormone to accelerate the metastasis for a cancer. There are 5 basic types of pituitary adenomas. The treatment is usually a transsphenoidal surgery for the removal of the adenoma. The detection is usually by MRI or CT scan. Visual problems may also be a symptom. Here you see some of the MRIs. You can see some of the mass here, where it's gone from a normal pituitary in the center to a microadenoma, to a macroadenoma, to eventually an aggressive adenoma. And it can attach to all the different cell types that produce growth hormone, prolactin, corticotropin, thyrotropin, etc. Let's take a look at a little more detail here. If we talk about prolactin cell, it's a lactotroph adenoma. Growth hormone cell, it's a somatotroph adenoma. Thyroid stimulating hormone cell, that's a thyrotroph adenoma. ACTH cell, that's going to be a corticotroph adenoma, and then a gonadotroph cell adenoma. Pituitary adenomas appear in the population about 16.7%. Roughly, we're talking about 800,000 a year. Okay? And with it comes the overproduction of stimulating or trophic hormones. The most common is the somatotroph adenoma. 27% of these pituitary adenomas, they will produce growth hormone, which is normally pulsatile. You can't really assay. You can assay IGF-1 and then compare it to stable levels, etc. You're going to look at acromegaly, excessive growth bones, and soft tissues. The next common one is corticotroph adenoma at 8%. It will cause the production of ACTH, which will in turn stimulate corticosteroids. This is Cushing's disease, which will be discussed later in the adrenal diseases. And then in between them is basically the prolactoma, which is basically the prolactin secreting adenomas. And you're going to see this by elevated prolactin levels. Of course, you always would use MRI to confirm. You can stabilize the patient if it's small using a dopamine analog, bromocriptine. If it's large, a transsphenoidal adenomectomy. And you want to monitor at all times the prolactin levels. To the lesser extent, FSH, LH adenomas, only about 5% of those pituitary adenomas. They produce causing increases in FSH or LH. They can produce gonadal hyperstimulation, which produces high estradiol and testosterone. One of the last ones that's common is thyrotroph adenoma, which causes the increase in TSH, which of course would stimulate thyroid hormone release. And this is called secondary hyperthyroid condition. And it will be discussed again a little bit when we get into thyroid diseases. But in other words, this is not the primary cause for hyperthyroidism. It's a secondary cause. There are also those non-functioning adenomas. And mostly they start off as a gonadotroph. They're about 32%. Others are about 9%. Okay? This is a transsphenoid surgery. Yep, going up through the nose in the nasal area. And what you do is you pass by the turbinates in the nasal cavity. You are going to basically go through the sphenoid sinus till you get to the floor of the sella turica, which is right here. This is the pituitary adenoma, and with an endoscope, you can basically remove that with, I would say, a relatively high success rate. I wouldn't say it's easy, but there are experts who know how to handle this, and it's been done fairly frequently. There usually is very little when we talk about removing this material, okay? And this is the best way to do it, because really you're going underneath the brain to go at this pituitary adenoma. We will have case studies this week because this kind of started off a little bit later than I would have wanted. All the case studies we'll review on Thursday, but you will have your case study document to be completed for Friday by midnight. Next week, we go through exam 1. This is going to cover chapters 1 through 3 in the textbook, plus all the lectures.